Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to continue with Standard 12, Chapter 11, that is Biotechnology Principles and Processes. So far we have learnt about all the principles of biotechnology. Then we have also learnt about the tools of recombinant DNA technology, that is the restriction enzymes. Then we have learnt about what are palindromes. Then we have also learned how different restriction endonucleases cut the DNA fragments. So in today's video we are going to learn about the separation and isolation of DNA fragments. So let us begin. So biotechnology principles and processes, separation and isolation of DNA fragments. Now the restriction endonucleases from DNA fragments that are separated via the gel electrophoresis. Okay, so whatever DNA fragments which are cut using the restriction endonucleases or the restriction enzymes are separated using the technique called as gel electrophoresis. So this is the setup of gel electrophoresis. So there is a power source and there is a glass container kind of, in which there is electrolyte solution, a glass slab, then the sample wells in gel. So there is a gel piece over here in which there are wells where the samples are loaded, and then there is this connection. So these DNA fragments are negatively charged. So what happens in this gel electrophoresis is that the DNA fragments which are cut are negatively charged. So these are separated by moving them towards the anode under an electric field via a matrix or the medium. So the medium used is the gel. Then most commonly used matrix is the agarose, which is a natural polymer extracted from the seaweeds. So the fragments of DNA are separated as per their size via the sieving effect provided by this agarose gel. So when the electric current or the power source is turned on, the fragments of DNA will move from the negative this thing towards the anode. Then how the fragments are separated as per their size. So the smaller DNA fragments which are there, the smaller ones will move the farthest, whereas the larger ones moves the least and will be typically near the loading wells of the agarose gel. So as we can see here in the picture, this is the typical agarose gel. The DNA samples were loaded over here in these wells. Here the DNA fragments were loaded. Then they will start moving from this end to this end. So the smallest ones have moved the farthest, whereas the largest ones are near the wells. Then the separated DNA fragments can be visualized. Okay, now after doing this gel electrophoresis, these are not visible to us, but they can be visualized after staining them with the ethidium bromide. So we have to stain these DNA fragments with ethidium bromide and then expose it to the UV light so that we can see them. Now after doing this staining with ethidium bromide and exposing to UV light, the DNA bands appear bright orange in color. Okay, so as we can see here, they will appear bright orange in color. Then the separated bands of DNA are separated from the agarose gel and extracted from the gel, and this process is called as elution. So what is done is, after the bands are separated, this gel piece will be cut, and then the DNA fragment will be isolated from that gel piece. So the DNA fragments, after this elution, the DNA fragments get purified this way and is used for recombination. So let us see again what is elution. So the process of cutting of the required separated bands of DNA from the agarose gel and extraction of DNA from the gel piece after gel electrophoresis is called as elution. Now these purified DNA fragments then is used in constructing the recombinant DNA by joining them to the cloning vectors. So this is all in today's video. In the next video we are going to learn about what are cloning vectors. So hope you have understood this. Thank you for watching my video. Please like, share and subscribe to the Modi Academy.